Okay, thank you very much, Dr. Rizwan. Dear teachers and colleges, good afternoon. Thank you for your attending to these very nice webinars. And I would like to invite Professor Budi. I think we already have him in this screen. Hello, Professor Budi. Good afternoon.
Good afternoon. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. So I will introduce Professor Budi. So Professor Budi will give us the insight about the role of genetic testing in vitro fertilization. So he has now he is a professor in obstetric and gynecology department, and he has a magister of public health in from Universitas Gadjah Mada Indonesia, and he graduated from his doctoral program from medical faculty of Universitas Indonesia. And he has reproductive endocrinology infertility consultant from medical faculty of Universitas Indonesia. He is very active now in reproductive system and health technology science. So now he is a staff at Obstetric and Gynecology Department in Rumah Sakit Cipto Mangunkusumo, Jakarta, and he is the president-elect of Indonesian Society of Obstetrician and Gynecologists, and now he is one of the international affiliate of the College Royal Australian and New Zealand College of Obstetrician and Gynecologists, and he also the chairman of the National Health Technology Assessment Committee, Ministry of Health of Republic Indonesia, and she also has a vice director of Indonesian Medical Education and Research in IMERI, Faculty of Medicine of Universitas Indonesia. Okay, Professor Budi, screen and time is yours.
Thank you very much. Thank you very much, Dr. Ini, for your very kind introduction. So, ladies and gentlemen, first of all, I'd like to thank the Board of the President, Indonesian Society, and also Malaysian Society of Human Genetics, Professor Gunadi, Professor Sultana, and also Professor from Malaysia. So, I'd like to start sorry, I'd like to start our discussion this afternoon with the topics of the role of pgt, implantation genetic testing in vitro fertilization. Just to remind all of us that this technology actually is already being 45 years since the first test tube baby, Louis Brown, was delivered in Bon Hall, London on July 25th, 1978, and then Professor Robert Edwards was the pioneer of this technology together with Professor Patrick Steptoe. They received a Nobel Award in 2010. And right now, I think more than 10 million baby in the world has been delivered by this kind of technology. And according to the Patrick Steptoe mentioned, his quote is very interesting. In 1978, he mentioned that we are at the end of the beginning, not the beginning of the end. So I think many black box, yeah, many mystery that should be answered, and then the technology is one of the things very important to bridge the gap. If you talk about the pregnancy in IVF, what is the mystery from gamete to the heartbeat? What is the missing link? Actually, if you think about the implantation failure, the failure of IVF, 72%, it happened because of implantation problems. And then we can see from the embryonic point of view, and also we can see from the endometrium point of view. The IVF failure was happened because non-implantation of the right embryo. So the good embryos, the normal genetic embryo is not implanted because the endometrium was bad. Or the failure is happened because the implantation of the wrong embryos. It means the abnormal chromosomally abnormal embryos was implanted in the very receptive endometrium, and then the pregnancy will be end with the miscarriage, will be end with the blighted ovum. That is for example. And then this is the Venn diagram showing us that 78% until 80% of reproductive failure is responsible because of the egg quality and derived by the sperm and also embryo and the womb. If you look these tables, it looks very interesting that the women more than 38 years old, they will have more than 75% abnormal embryo chromosome. So that's why the success rate declining very sharp after the female more than or above 38 years old. And this table is quite interesting, I think, from Santiago Muno from Colorado. It's about more than the data, almost 30,000 embryos showing us that euploidy rate, it depends on the age of the mother. We can see if the female below than 35 years old, 60% embryos is euploid. But extremely, for the female above 42 years old, only 10% embryo is euploid. So that's why it's not so easy, yeah, to get the pregnancy for the women over 40 years old because the euploidy rate is very, very low. And then how the role of the sperm? The sperm also take a very important responsibility because if the number of the sperm is quite low, less than 1 million, yeah, the sperm has 30% of abnormal in chromosome. This is being based on the sperm aneuploidy test for 13, 18, 21XY chromosome. So we can imagine the patients with the very bad sperm, of course, it will increasing the risk of the embryos becoming aneuploidy. And then this is the data coming from the patients who underwent ART and then suffering from miscarriages. This is the IVF, almost 63% if miscarriage, the product of conception is abnormal. 63%. And then if the patient is the sperm less than 5 million, almost 73%, the product of conceptions is abnormal in chromosome. It's quite different with spontaneous pregnancy. It's a little bit significantly lower, and also compared with egg donation, which is the egg donation is coming from very young ladies. So that's why very rare we found the chromosomal abnormal problem. So that's why the precision reproductive medicine is really promising. We should select what is the best sperm, what is the best egg, and then what is the best embryos that you want to transfer to the womb. So that will lead to the pregnancy. So one of the methods of selecting embryo is preimplantation genetic testing. As we mentioned since the earlier, that this is the genetic testing of embryos that we can do through the polar body biopsy, blastomer biopsy, trophectoderm biopsy, or blastosynthesis to obtain embryonic DNA. This is a very important how we obtain embryo DNA to the biopsy. Yeah, we can compare later. And this is actually the biggest challenge. The biggest challenge in preimplantation genetic testing. When the first time when the Yuri Verlinsky, the father of PGT, that one previously we mentioned as PGD, preimplantation genetic diagnosis, he biopsied the polar body, the first polar body biopsy. It means only the maternal part of the chromosome can be detected. And then later on, the technique was developed. We do the biopsy from the 8 cell embryo, blastomer biopsy, and the last trophectoderm biopsy when the embryo was on day 5. And then later we talk a lot about non-invasive preimplantation genetic testing. This is only for a few data this year in our center. We did a pgt-a from 77 women. And then let's see, this one is almost the same. The patients coming from 35 or below or above 35 years old. Then we can see that chromosomal aneuploidy was present 63% the above 35 years old. for the female. And then this is one 234 embryos. This is the chromosomal aneuploidy. We have 45%. We have monosomy, we have trisomy, we have mosaic. Even the mosaic is quite difficult to be detected through the pgt-a. It's not so easy. And then the sex chromosome as well. We have also some abnormalities in the sex chromosome. So the next question is, right now we are in the era of non-invasive preimplantation genetic testing as the next revolution. If we compare before we do the biopsy, that has a risk to make an injury to the blastoma or the pectoderm. For example, yeah, we do the biopsy, which is infective and then much harmful to the embryo, and also they should have it needs very special equipment for the biopsy, the laser to facilitating biopsy, embryo biopsy by embryologists, and also laboratory and also the skillful embryologist is very, very important. So that's why now the people trying to improve the method, yeah, to less invasive. Yeah, this is the how the embryologist working in the lab. This is the embryological laboratory lab. This is the micro manipulator with the laser. If she wants to do the biopsy, she put laser thinning the zona pellucida and then penetrate the zona pellucida, trying to biopsy to make a biopsy of the tropectoderm. Yes, now the people with the non-invasive preimplantation genetic testing try to investigating the new sources of embryo-derived DNA, which is the previously we only have the DNA from the biopsy. So this is, for example, the invasive PGT DNA could be extracted from the embryonic cell. This obtained by the DHB embryos. We did the biopsy here. There's one or two cells or this one, the five embryos, the embryos with much more cells. We did the biopsy, the tropectoderm, or we do aspiration of blastocyst fluid in the day 5. And then this one is totally non-invasive. We only investigate the spent culture media. Spent culture media, we do isolate the cell-free DNA that we can find it in the spent culture media. This is very important, and this is really, really non-invasive. So as summary, this is all the method how preimplantation genetic testing did in our IVF lab. This is the previously done the first polar body biopsy, blastoma biopsy. This is tropectoderm biopsy, blastosal fluid biopsy, and then now I think it's quite looks promising. It's spent culture media. This is really invasive, really non-invasive, but the big question only the confidence, the cell-free DNA analysis with the tropectoderm biopsy in terms of the chromosomal analysis. So this is the evidence how the people trying to establish the spent culture media. They did the collection, timing the collection between day 2 until day 5 of embryo culture. And then make some in time incubations, and then interestingly that the cell-free DNA can be isolated from the spent culture media. It looks promising in terms of concentrations, ranging on average 18 until 70 nanogram per milliliter, which is sufficient enough to perform NGF. And then this is the debate surrounding the efficacy of non-invasive pgt-a. The amplification rate is ranging between 80 to 100%, and then the ploidy concordance rate this also looks promising. The average is 80%, comparing tropectoderm biopsy with cell-free DNA in spent culture media. Only one or two may be extreme results. So if you look this one, this is all the benefits is coming from spent culture media, PGT, the non-invasive, easy, safe, and all embryos could be assessed. The concern only if we found the low cell-free DNA concentrations, and also how do we control DNA contamination, and how do we convince the origin of cell-free DNA. And this is all the limitations, low sample size, and many things that still restricted evidence. This is the origin of embryonic DNA in culture media. Actually, the human blastocyst, the cell number, that, and allocations during the late Preimplantation development in vitro during the culture, there will there was a widespread cell death in the body of a tropectoderm and inner cell mass. I think so. That's why we did the biopsy tropectoderm. It will representing at least 80 to 90% of ICM. So if we do analysis of tropectoderm, it means 80 to 90% ICM will be represented by doing biopsy. And then this is the origin of the cells is coming both coming from apoptosis or necrosis. This, for example, this is a programmed cell death. This is uncontrolled cell death between 70 to 200 base pairs, and then this is cellular feature. What we did through the PGT by PGT. And this is one of the piloting in our center. We are trying to develop pgt-a from the spent blastocyst medium culture. Firstly, we do the collection 22 spent culture media from blastocyst, and then we pulling and then running the sample with MySic NGS. This is the 37 blastocyst stage analysis. This is around 20 microliter each drop, and then 2.5 micro for amplification, and do the sequencing with Illumina MySic NGS. This is the spent culture media, and then the control, we do the topectoderm biopsy on expanded blastocyst on day 5 embryo culture. And then this the result, we can do 100% amplification rate, and then this is euploidy concordance, and then aneuploidy concordance, the sex chromosome concordance, and the total chromosome concordance between spent culture media and topectoderm biopsy is about 82%. So it looks promising, the first piloting study. And then this is the data. This is the NGS result. This is from the topectoderm biopsy, monosomy 21 XX, and it's the same, exactly the same with the spent culture media from blastocyst. The result is exactly the same. So the concordance is promising. The risks associated with non-invasive PGT-A is the DNA contamination. This is very important. So that's why we have the mitigation, the proper measure to prevent DNA contamination. And then also the clinic, the IFF clinic, very important to change the culture procedure because could be one embryo, one drop. We cannot do the group culture because the contamination coming from if we culture model one embryos. Yeah, this is of course the promising, the ideal clinical non-infective, this robust, high sensitivity and specificity, reproducible, workable in all clinic, and then looks cost effective because we did not need special equipment like a laser for the biopsy of the tropectoderm. And the next also we do the validation study for non-invasive PGT-A for about 240 samples. This is we're comparing the single culture media with two-step culture media. It means we do the collection once in the day 5, and then the second group we do the collection twice in day 3 and day 5 of embryo culture, and then we compare with the topectoderm biopsy. Yes, this is the result. This is the embryo. The average of daily concentrations coming from single culture media. This is two-step culture media. Then it's coming from topectoderm biopsy. Yeah, it's not significantly different. Then we can do the 100% amplification, all the coming from the topectoderm biopsy, single culture media, and then two-step culture media. And then all can be interpretable by NGS. The total euploidy and aneuploidy is almost the same, but still we have some disconcordance. This is, for example, this is full concordance. 46 XY. This is, for example, partial concordance. Yeah, this is the data from topectoderm biopsy. This one from the spent culture media. This is false negative. In the spent culture media did not recognize any abnormality, but however, in the topectoderm biopsy, we found any abnormality, and this is a false positive. In the spent culture media, we found one abnormalities, 47 plus 16. This is no abnormalities in spent culture media. So for overall, this is in a single culture media, the sensitivity about 98% and 80%, and then two-step culture media, this is the 85% for sensitivity and 70% for specificity. So this is it looks promising. Another thing also in we do the study for microRNA. So instead of cell-free DNA for Preimplantation genetic testing, also we are trying to see how the microRNA in spine culture media in order to detecting there is some abnormality in the chromosome is coming from the embryo. We know this is the role of microRNA. It's very important, and then this is microRNA also span in the culture media. And our microRNA is very important in keeping role in communication between parental and embryos, and then inside also microRNA is very important. This is microRNA in the human span media culture embryo. Many microRNA was identified in the human span culture media right now. Human blastocysts express many microRNA, and then this is known to be separated from the cell culture. And then it's very important how the microRNA play their role. This is repressing translational, stimulation translational, and do also RNA interference. This is non-coded RNA, we know, but it's very important how microRNA also uh, keeping the chromosome still normal. So it will be increased if they found any abnormalities in the embryo's chromosome. So this is our study. Again, yeah, we are trying to see how the microRNA we can be used in the spine culture media to detect any abnormalities in embryo. This is the data from 36 embryos. We do the single cell culture, and then we do the tapetobiome on day 5. Then again, this one is a spine culture media. For tapetobiome biopsy, we use the NGS MySeq. And then we use this one, we use the PCR to see how the microRNA. Then we found the microRNA 135B expressions in human blastocyst culture media significantly higher when there's an haploidy. Yeah, this one, the microRNA 135B, yeah, compared to the euploid embryos. Yeah, this is one of the microRNA. Another microRNA also we check. This is, for example, the sensitivity and specificity of microRNA 135B. The spine culture media looks, to of course to be enhanced in the research. Also, we published how the microRNA 135B expression is increasing in older age, in older women, that is positively correlated with the chronological age and also biological age. So that's why 135B looks promising if you want to see to detect any embryo chromosome, abnormal chromosome of embryos. This is compared with non-invasive pgt-a from Pentastar media. This is euploidy XX, this is aneuploidy. So I think no difference. So this is the golden standard. We use the cell-free DNA by MySeq. Then the compare with microRNA also is running very well. And the other thing also, microRNA 372 and microRNA 661 also detected and then increasing 1.6 fold higher in media coming from the ploidy embryos than media from the euploid embryos. And then this is also statistically significant. And the last, microRNA 661 also is 1.5 fold higher in media coming from the ploidy embryos. So that's why it looks promising right now. People trying to establish non-invasive pgt-a in order to improving the quality, improving the access, uh, and then decreasing the risk to do the biopsy of the embryo, particularly for the corpuscular biopsy. So I think I'm coming to my last slide. So as the take-home messages, the effectiveness of the new approach right now, particularly for non-invasive pgt-a, has potential for development chromosome screening. The applicabilities, as Professor Gunadi mentioned, very important early detection and non-invasive. Yeah, we don't discuss now about the intervention. How could we make some intervention if we found any abnormalities in the chromosome? Yeah, let's say we only talk about pgt-a, we still have pgt monogenic disease, pgt for structural enhancement, and then pgt for polygenic diseases. And then the next also very important, the origin of the cell-free DNA is coming from stem cell media. Yeah, we should convince. And also this is coming 100% is coming from the embryos, including if we measuring also the microRNA is very important. And the lack of both well-designed studies are required to increase the number of evidence, right? Person, non-invasive pgt, yeah, still looks promising, but still recommended with together with the trifecta biopsy as a golden standard. I think I come to my last of presentation. Thank you so much for your kind attention. Thank you, moderator.
Okay. Thank you very much, Professor Budi, for a very insightful and interesting talk. I think every obstetric and gynecologist and also pediatrician dreams to have a programming a healthy baby and do early detection of minor abnormality from an embryo. So maybe I would like to do let me do a little wrap up from Professor Budi talk that nowadays we have non-invasive prenatal testing only by taking the culture media seems promising, and Prof. Budi has revealed from his lab that this NIPNT has higher sensitivity and specificity compared to invasive one, and we can see a very rich hybridization product from the PCR. Okay, Professor Budi, maybe we can go to the next speaker, Dr. Rizwan. Thank you so much, Professor Budi.
Thank you.